I've heard Dr. Light speak before. It was one of the most inspiring talks I've ever heard, and I was happy to hear parts of it again. But my question, I wondered if you could speak a little bit to an example you gave in an earlier talk I heard about the clinical response of GI infection in mice to stress. That was really impressive to me, where you stressed the mice and you saw a completely different response to a GI infection.
Yes, I didn't present that here because there's a lot of the gut infections. And basically, the more you stress an animal, the bug... Oh. Okay. Yeah. There's more and more papers coming out now, and we have some out. There's other groups that have others where we're doing a lot of sequencing now. So we sequence the whole genome that's present, the whole microbiota. And it changes, what we're seeing now is that any type of stress changes the community of organisms and the prevalence that's present in the gut. And what happens is that there are then very much reactive species within the gut that then decide to come out. For example, E. coli being one of the most prominent there. So the effects of stress within the gut is such that there is a complete changeover in the community. The community then becomes more infective, and you start finding bugs now not hanging out anymore in the lumen where they used to want to hang out. They're now starting to attach onto the epithelium of the gut and starting to want to invade. They start to now express different adhesive properties, adhesins, and different pili, which they didn't express before, but they will express when they see the neurochemicals. So with all this, we really don't understand what's going on, because what's missing in a lot of this is everything regional. Just like in the lung, you can say, well, I'll measure the blood level of a neurochemical. That doesn't tell us a lot. It just tells us it's there. What is the regional level at the surface that you want to look at to say, is this bug going to invade and cause disease there? That all those things are yet to come and have to be done yet. But stress as itself is a factor which the bacteria recognize, and then they institute their own evolutionary program how to deal with it. Does that?
Currently, there are several publications out showing there are different types of stresses that we go through in life, and they rank them as good, bad, and toxic. Do you have any way of quantifying that and applying it to the animal?
Oh, that's a great question. I often put, what's the stress of someone in a motor vehicle accident compared to somebody, when I used to be in Minnesota in surgery, saying that, well, I'm shopping at the Mall of America and I forgot where I parked my car, and then I'm stressed out like crazy, and 2 days later I come down with a cold. Was it the stress that did it? I think that's a question for psychologists and the rest. I think I tend to deal with both psychological stressors and physical stressors. I think that the psychological stressors are a bit different. I mean, what is stress now compared to someone or an animal in the 1800s that had to deal with a completely different environment? Don't have a good answer for you in that. I think what we have to do is get down on the level of the organ where we think the infection occurs, and says whatever stress it is, we need to quantitate at that level. But I would think a physical acute stressor is something that is going to increase the immune system, and yet the bacteria respond to it. Chronic stressors are different. I think this would take like an hour to answer, but I don't think there's any gradation yet. I don't have a good answer for that.
Mark, as usual, another great talk. But I want you to add another factor to this, and that is the other thing that we see besides stress are tremendous dietary changes. So in terms of the substrate now for the bacteria, either we have animals that have a long haul, so their GI tracts are empty, or we make tremendous changes from refuge diets to concentrate diets. So we change the media that bacteria has to grow in. How do you see those things interacting?
That's a great question. I've left out that whole side of the talk, which is that everyone keeps forgetting that all the food you eat has neurochemicals, and that the neurochemicals go all the way back. I mean, I had a slide I took out which was potatoes have norepinephrine. Anybody grow tomatoes out there? When they get cold stress, they make norepinephrine, and they actually can get infected. And so tomatoes, it's been published, make an antibiotic, which is the conjugation of norepinephrine or dopamine to another moiety, and that actually keeps the infectious burden down. But silage that you use for your cattle contains large levels of tyramine, another neurochemical. So what are all the neurochemicals in the food? Nobody quantifies it. I once did something with one of the food companies, I won't mention which I took their rice, corn, raisin, varieties of their breakfast cereal, and I thought I would have a neat grant. And what I did was I ground up the cereal, I looked at the neurochemicals, I then put it with 0157 that infects children, and I had the vice president for research for this big multi-billion-dollar company in my lab, and I said, hey, look, your corn stuff is more infected because of neurochemicals and makes 0157 grow that affect kids more than your rice one, more than your raisin one. That person turned white on me, walked out, and didn't want to have anything to do with me. And I said, hey, you can understand then your sources of your grain and what it is and promotes the bacteria in the gut. Shouldn't you be doing that? I never heard from them again. So I think the way we look at food, we don't quantify any of that, and I think we should.
Mentioned the change in antibiotic resistance, and it took me back to the 1970 study of joyriding pigs, where they took pigs from farms that never had an antibiotic on them, took them for rides in trucks, and came back, and the resistance profiles in their gut flora dramatically changed. So the question to you is, when you're talking about resistance, is the evidence primarily pointing towards change in populations with overgrowth of previously minor populations, or is it actually speaking to maybe the ability to affect turning on or off a gene?
I think it's speaking to both, but a lot to the latter. And studies by other people have shown, again, increasing conjugative transfer between bacteria, actually throwing over more antibiotic-resistant genes because they've been stressed from one species to another. We've also looked at bacteria when they've been put with norepinephrine and other neurochemicals, and we've looked at their antibiotic resistance. It changes as well. You make a tougher bug. Remember something really important here. Anybody who grows bacteria and grows them in the laboratory on a petri dish and grows them in TSB is a great life. You know, that's all glucose, all nice and happy. That's not the way it is. It's a tough life out there for a bug to grow in the gut or the lung. Everybody's trying to kill it or move it over in different stuff. And, you know, the way I put it this way, I mean, if someone came down from outer space and picked up a human who's been eating all day, every day at an all-you-can-eat bar and weighs 400 pounds, that's a Homo sapiens. And if you go and get someone who's not had a lot of food but has six-pack abs and a Kalashnikov and is trying to survive out there, that's a Homo sapiens. But they've had two different experiences. And so when we grow our bugs in the laboratory and we grow them nice and easy all the time, it's been known, and I have a paper from 1918, that that bug isn't the same as the same bug which is passed from animal to animal. They are the same bug genetically, but they're not the same bug when it comes to infection. It's a tough life. The bugs respond different. They're hardened out there, remember.
This is for Dr. Claussen. All of the topics today were kind of high-tech, and yours especially, but basically you touched on at the end of your talk, when you get the wheels on the ground, what do you see out your windshield? What direction are you headed now?
Well, there's two. I guess I have two responses to that. One is, what is the host effect to mHeme, for one? So I just talked about typing mHeme and how those clades shake out. What we didn't talk about, and what we are doing, what my colleagues back at the research center are looking at, is what is the host resistance or susceptibility to mHeme? Is there any genetic allele there that can be used to help manage BRDC? So that's one. The other, so if, again, this is preliminary data. If it does hold up, and I think that it probably will, then the leukotoxin gene, I picked that gene just because it's a major major virulence factor, but that signal isn't just in the leukotoxin gene. It's all throughout a lot of the genome. And so now the question is, what is the block on the mHeme genome that contains that signal, and what genes are at play in there, and where does the true association for that kind of behavior lie, and can we develop tests to potentially exploit that? I mean, can we use, aside from the host effect, on just using the strain, can we use strain typing as a way to potentially identify cattle that are carrying those bacteria that have a high propensity to cause disease if the environmental circumstances are correct? Can we identify those cattle and get them away from the rest of the population? And that remains to be seen, and an important component to that is frequency. What is the frequency at the SMP level, which is a little high resolution? With SMP level, what is the frequency of strains out there that have that genetic predisposition? And if it's a low frequency, then perhaps we could use it. So I would say exploiting that information to dial into the biology behind it, for one, is important, and then potentially using it in the field to perhaps as a tool to prevent outbreaks in some situations.
You hear that? Okay. Dr. Light, I'd like your comments on two different scenarios. Have clients in California, Nebraska, and Oklahoma who will bring in long-haul calves direct from the ranch, not preconditioned. They're vaccinated once, turned out on the hills, so they go right back on grass, and see a much, much lower incidence of morbidity and mortality compared to ones in the same scenario that are brought right into dry lots, into backgrounding lots, and go that way. So the difference in the gut level of the feed that they're receiving, grass versus a feedlot ration. The other scenario is have one client in southeast Idaho who for several years has weaned 6,000 to 10,000 calves for himself and other people. And last year, the one difference he did differently was he went to using a new dart gun, treating the calves right in the pen, not taking them out of the pen like normal and into a hospital pen. And he reduced his realizers from over 60 down to 6, and his death loss, which is already very low, he cut in half by simply treating the calves in the pen, leaving it in the pen with a new dart rather than taking them out. So just your comments on those two different scenarios, why we see such a big difference in morbidity, and in the other case, a big difference in mortality.
So let me understand. The second part is when they actually immunize them in the pen and don't move them out to the hospital to do it, he's seeing a lower amount of morbidity, correct?
Well, he was seeing the same level of morbidity, but when he treated the calves with a new dart gun in the pen,
right,
didn't take them out, his response to treatment was way higher, so he had fewer chronics, fewer realizers, and cut his death loss in half.
Right. Well, in that case, what I would say, he's keeping the social environment that the animals are in and what they recognize as the same, and he's not changing that. So it would seem to me that their stress level is going to be less in their home environment than moving them out to the hospital and then giving the dart. So you're having less of a stress response of the animals at that point. You're probably at that point affecting, since you're having less of a stress response, less of a bacterial response wherever in the animal, and at the same time maybe the immune response is also a better response at the same time as well. In the first case was the feeding having a higher amount of mortality, I believe, or morbidity when you fed them when they were fed in the pen, when they were brought in, versus being turned out and to be fed grass again, correct? Right. So in that case, again, the diet is going to shape the microbiome to a degree that we do not yet appreciate enough, whether it's clinically or on the farm side. And so those bugs that the animal have had established its microbiome in the gut, which is what it was founded with, it grew up with. When you abruptly change it then, when it's in the pen to the hay, I'm sure you're changing the whole distribution of the microbiota that's present at that time. And that's going to have consequences that, in that case, seem deleterious to the animal. Returning it to grass again is going to allow those bugs that had populated the animal knew about to stay there and repopulate themselves and to establish a healthy gut. That's what I would think at that point is going on.
Well, to me, it's just really interesting as far as our management of high-stress calves, where we have this situation of in different parts of the country, they bring them in, vaccinate them once, and kick them back out on grass, that their morbidity and then their mortality, if they do have to treat something, is just way, way lower. Like the instance in Oklahoma, they'll be bringing in those Missouri, Arkansas calves. They do go through a revac program in that situation, but the ones that go out on grass traps, in the end, get along way better. If they keep buying calves, they fill up their traps and have to start confining them to the pens, then their mortality rate goes right up.
Just remember something. It's not only what the bacteria want, but it's also what the brain wants, whether it's cow or human. And I'll bring it back just for a second to the human side. Most of you may be familiar with what's known as antibiotic-associated diarrhea in patients. When they get high-dose antibiotics for a long time and they get antibiotic-associated diarrhea. Here's my question. And I've asked every GI guy I know about this and every GI woman about this and never have gotten a good answer. Okay, now you changed over the whole microbiota, you've given all these doses, now you got a completely changed microbiota. The patient then goes back to eating normally. How does it know what bugs to bring back? What's telling it to reestablish it to what occurred before I gave the microbiota? And what I've posited is the brain. The brain is telling it what it wants because there is a communication circuitry going on between the gut and the brain and the gut. And so feeding the animals back on grass is bringing back not only the nutritive sources that the animal wants for the bacteria, but then that's communicated to the brain. The brain communicates back. Here we're going to establish the population which is most beneficial for us. So I think there's a lot to be said for the design of different diets, what I've called almost as in a grant I had, and they got trashed totally because they thought this was just totally off the wall. As I think Chris knows, a lot of my stuff initially had been 25 years ago, totally, which is I called it dialing in the microbiome. Can you design feeder diets for your animals that gives it the profile you want, not only in the microbiota, but also its brain? Because then you're having a synergy going between the two, and you have happier animals. Better from a different standpoint. But I've tried that with some people saying, hey, we could design these, and we have a paper coming out now on this, that you can actually design that to give you a microbiota that then gives you a different type of psychological profile on the animal.
So could you possibly comment on the role of probiotics in cattle as a way, not only in the gut, but is there a possible application of probiotics in the nasal pharyngeal region as a way to maintain diversity that might cut down on pathogen overcrowding?
Oh, I definitely think there is. I mean, the whole probiotic field, definitely, because remember, the probiotics are making neurochemicals all the time. They make all these panoply of things that we never consider. And the functional food industry knows about that. They actually make breads enriched with GABA that have come from lactobacilli. That's the source. Soybean milk is enriched with lactobacilli making GABA. So yes, you can engineer your strain to make whatever neurochemical to help establish a certain microbiota profile. That definitely could be made. It's been proposed by other people already about that. And in terms of the brain, there's a whole field now. For example, there's a whole elementary pharmabiotics center in Cork, Ireland, as well as, if I pronounce it right, Chegis, the animal facility. They're all doing this stuff as well. And about 3 years ago, I put out the thing that probiotics are really drug delivery vehicles. That's what they are. Everyone looks at them from the immune standpoint, but they're actually drug delivery vehicles because they're making neurochemicals. We don't know why they're doing it totally, but they make it, and then we have to then balance it with the food source that they give at the same time. We may have a new way of using it for cattle and the rest based on that.
Okay, I guess my question almost became irrelevant because I was going to suggest that Dr. Claussen and Light start talking to each other, which seems to have happened. But I think, Dr. Claussen, in terms of what you told us about clades, lung clades and that, I think it would be very interesting to have you and Dr. Light address that, because you've done sequencing and shown there are very different clades in the lung versus the upper respiratory tract. I don't know how many colonies you picked to see that difference, but based on what Dr. Light has suggested, those clades were already there in the upper respiratory tract and then sensed these changes, and there was a selective invasion. But I would like to hear the two of you address that issue.
Well, I agree with a lot of what you said there. I will throw out. I do want to be clear that honest about this. You know, the data that I showed was one isolate per animal. Okay, so those sick animals, one isolate came from there. The upper respiratory tract of the healthy animals, when we got those samples, we plated them out. We have multiple samples, multiple isolates from each of those animals. The tree that I showed was just one colony, one isolate from those samples. So bear that in mind, that the issue of how many strains are really in the upper respiratory tract, is that the only one? I can't say it right now with the data I have in hand. So I just want to throw that out there, that data was limited in that way. So, but certainly there can be more than one strain present in the upper respiratory tract. I mean, people have already showed that with pulsal data that's out there. There's not just one type. So, yeah, they already are there. And the interaction of these substrains of M-heme with the host in terms of the signals that they're sending to one another, the excitation that the bacteria might get from the host as a cue to potentially gear up its protein profile to get down to the lung. Absolutely, I think that's in play. So in terms of substrains interacting with the host, signaling between the host, getting cues from the host to behave in a certain way, yeah, absolutely, I think that's online.
I think all I'll say is that I think what the other speakers have brought up is critical, is that we assume that if we have M-heme on a plate that all the bugs are all the same. They're not all the same. They're a responsive population. And what does that mean for infection? What does that mean for their response to be able to produce? We don't know. I mean, one time I tried picking out individual colonies. Yes, they have different abilities. But when you try to put a grant in NIH, it's called fishing. You can't do that, and it would take forever. So I think there's a lot of potential for that, to understand the synergy that's going on between the different bugs, because there is a synergy going on where they are existing as communities together, and they're signaling back to themselves or signaling with us. And I just think the realization that one strain of M-heme or E. coli that we get, and you talk to sometimes the molecular people, and they say, well, it's all the same. We can understand everything. And you say, no, I can grow that strain and see different colonies appear. They're like, we don't want to know. Sometimes we don't want to know about that, because then they have different phenotypes.
One thing I want to say for this Kansas State study that is being led by Mike Apley, one of the things that we're looking at is not just M-heme, but the total bacterial diversity that's in the nasopharyngeal regions as animals go from a cell barn to a feedlot, and different treatments that they undergo. And so it is a complicated question. What you have on a plate is in no way, it's absolutely right, it's no way reflective of the true biological state of microbes. Most microbes want to exist in biofilms. They don't want to exist as a colony on a plate or growing in a culture being shaken at 37 degrees. That's not their natural state. The biofilm is. And so, but that is one of the questions that's being asked in a grant right now, is the overall diversity in the upper respiratory tract, and how that diversity interplays with what happens in terms of the manifestation of disease. Layered on that is more complexity, which is the host itself, the signals the host is sending to that community and getting a specific outcome from that.
With that, I think we're going to... Yes. We'll take one more question, and after that, we'll go to our morning break.
Okay, thank you. You didn't really talk about this, but on one of your slides you did mention the pulmonary intravascular macrophages. And so I was wondering, has anybody started to look at that even histopathologically in BRDC lungs? I mean, everybody talks about the alveolar ones because you can get them out in BAL, and the PIMs are harder to pull out. So just curious.
Yeah, and Carol, I think that's the big problem is it's tough to isolate those. But if you do IHC, and you look at some of the markers on those, they will respond equally well, if not better, than the alveolar macrophages. So I included them as one of those liabilities from the standpoint that if you have endotoxemia, who do you activate first? Those intrapulmonary macrophages or the alveolar? And I would say it's the intravascular. So as we talk about vascular leakage, increased permeability, and that tremendous effusion of fluid and fibrin that occurs in the early stages and may then actually inhibit bacterial clearance. Maybe it's those pulmonary intravascular macrophages are more the source of that change than the alveolar macrophages. So, yeah, they may end up being more of a liability than an asset in some cases. So that's why I kind of included those as part of that. Thanks.
Thank you very much. I think with that we'll take a break.